Hi, I'm David and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. In question 1, we have a diagram showing a chloroplast and some processes that occur in it. A are the membrane-bound compartments in chloroplast that make up the granum. They are called the thylakoid membrane. A2 wants you to explain how the structure and appearance of the granum and thylakoid relate to their function. You are asked to explain the structures. You cannot receive full marks if you only state the features without explaining how they contribute to the function. Many thylakoids provide a large surface area. This allows an increase in light absorption, leading to a higher rate of the light-dependent stage. The thylakoid membrane has many pigments that are arranged into photosystem. Numerous electron carriers form the ETC, as well as the enzymes required for photophosphorylation. These structures allow light-dependent stage to occur. You can also refer to one specific reaction related to the structure you mentioned. For example, if you talk about electron carriers, you can explain that they create a proton gradient for chemiosmosis. The aqueous space enclosed by the thylakoid membrane is the thylakoid lumen or thylakoid space. When electrons are passed along the ETC, protons are pumped into the thylakoid space. So, this is the part that allows the accumulation of proton. The thylakoid membrane and granum appear green under the microscope. This is due to the presence of chlorophyll. Chlorophyll is a green pigment. It reflects green wavelength and absorbs red and blue light for photophosphorylation. The metabolic pathway that occurs in the stroma of a chloroplast is the Calvin cycle. It is described as a cycle because there is no start or end point. All molecules are converted into the next one and the first molecule is regenerated. In other words, all molecules are present all the time since no one is used up as long as the cycle continues. In the Calvin cycle, the starting molecule, RUBP, is regenerated when a full cycle is completed. You can also get 1 mark if you provide a numerical detail of the molecules involved in the Calvin cycle. B is the product of photolysis, the process of breaking down water into hydrogen and oxygen molecules in the presence of light. D is the product of the Calvin cycle. Carbohydrates, amino acids, and lipids can be produced in the process. 2. Suggest and explain the importance of glucose and the product labeled in B in figure 1.1 to ecosystems. Although oxygen is considered a waste product in photosynthesis, it can be utilized in aerobic respiration. It acts as the final electron acceptor in oxidative phosphorylation. Glucose is a commonly used respiratory substrate. It is used in respiration for ATP production. The photosynthetic products synthesized by plants are involved in energy flow through food chains and food webs. They serve as the primary energy source for many other organisms in the ecosystem. 2a. Outline 2 other levels at which biodiversity can be measured. The first one is the ecosystem or habitat diversity. It is determined by measuring the number of different ecosystems or habitats. The second one is the species diversity. It measures the number of species and their relative abundance. Here, we have information on how genetic variation is calculated. B. Explain why scientists use databases and computers to calculate the mean number of nucleotide differences per site. The study involved obtaining DNA sequences from many individuals. A significant amount of data will be involved. Databases allow the storage of many DNA or nucleotide sequences from all the samples. We can also use software to align and compare these sequences. This will be faster compared to manual comparison. When we want to retrieve a specific sequence, we can easily search for it in the database. Additionally, it is possible to process, analyze, sort, and perform calculations on large quantities of data. Without computers, we would be unable to handle such enormous amounts of data. Sharing data is possible using a cloud system. This information can be accessed from anywhere in the world. Lastly, Software and AI can help us reduce the time required to process information. Table 2.1 shows the mean number of nucleotide differences per site of some species. Higher differences indicate a greater variation in the species. Anopheles gambia shows the most genetic variation. Its scientific name is composed of two parts, the genus and the species. The first word is the genus. The first two organisms belong to the kingdom Animalia. Plasmodium is a protoctist. Z. mace belongs to Plantae. So, there are three kingdoms represented. D1 give reasons why low genetic variation may decrease the long-term survival of a species. Genetic variation is the raw material for evolution. A population with low genetic variation exhibits few differences among its members and is less capable of adapting and evolving. This is because there is little variation for selection to act on. This means that most members are very similar. There are not many choices for the environment to select the best adaptive features. Without a great variety of values, there are few potentially beneficial alleles. Not many individuals would have a selective advantage. The same selection pressure, such as an infectious disease, could kill most members. The lack of unequal chances of survival resulted in difficulty in evolving. If the low genetic variation is due to a small population size, this decreases the species' survival chances altogether. This is because it is very likely that they will be wiped out entirely by a changing environmental factor. Figure 2.1 shows how the IUCN categorizes species according to their conservation status. D2 asks you to predict the relationship between genetic variation and conservation status based on the assumption from the previous question. If a low genetic diversity is assumed to decrease the chance of survival, the species would be more threatened with extinction. So, it should have a higher conservation status and is more vulnerable to endangerment. Figure 2.2 shows the mean number of nucleotide differences per site of some organisms and their conservation status. D3 wants to assess whether the data provides support for the prediction you made in D2. The greater the number of differences, the greater the variation. It was assumed that they were less likely to become extinct. Many of the species in the graph do not support the relationship. For example, chimpanzees have the highest genetic variation but are endangered. Minke whales have the lowest genetic variation, but they are the least concerned. These are the opposite of what we predicted. Some species exhibit similar genetic variation, but have different conservation statuses. For example, wolves and gorillas have similar mean number of nucleotide differences per site, but one is least concerned, and the other one is critically endangered. The gorilla is the most critically endangered of the species, but it has a relatively high level of genetic variation. There is a mark for AVP. For example, the data is limited to only 7 species. Therefore, it is insufficient for us to use it to support a prediction concerning all species and their conservation status. Other factors that may affect their conservation status, such as hunting and poaching, were not considered. 3a talks about a gene that determines the flower color of morning glory. The question wants you to calculate the number of heterozygous plants in the field using the Hardy-Weinberg equation. P and Q represent the dominant and recessive allele frequencies. P squared, 2pq, and Q squared are the frequencies of homozygous dominant, heterozygous, and homozygous recessive individuals. 440 out of 1,100 individuals are homozygous recessive. That makes Q squared 0.4. Then, we can calculate Q. P will be 1 minus Q. The frequency of heterozygous individual is 2pq. Multiplying this value by the total number of plants gives us the number of heterozygous plants. B suggests 2 reasons the flower color phenotype shows continuous variation. The question describes the flower color as a characteristic that can change gradually. This means the phenotypes are not distinct. We cannot record the color in different categories, and there is a gradual range. There are various intermediates in the phenotype. Besides, it's a polygenic trait. It is controlled by more than one gene. These continuous variations, on the other hand, are usually controlled by only one gene. Lastly, continuous variation is usually affected by the environment. The question mentioned that the carbon dioxide concentration of the surrounding air can cause a change in the flower color. In C, we have a graph showing the relationship between the carbon dioxide concentration and the mean width of stomata apertures. 1. Suggests how the stomata response shown in figure 3.2 would allow the plant to survive the effects of climate change. There is a negative correlation between the variables. An increase in carbon dioxide concentration causes the stomata aperture to decrease. Stomata are the primary sites of transpiration in plants. When the aperture decreases, less water loss occurs as the rate of evaporation and diffusion decreases. This allows plants to survive in low water availability or dry conditions by preventing excessive water loss. 2. Describe how abscisic acid causes the closure of stomata. Abscisic acid is a stress hormone that plants produce in response to challenging situations, such as low water availability and high temperature conditions. It binds to receptors on the gut cell surface membrane. They have complementary shapes. The binding causes proton to stop moving out of the cell into the cell wall. This is because it inhibits proton pumps on the cell surface membrane. Calcium ions will then enter the cytoplasm of the cells. They act as a second messenger. Channel proteins that allow potassium ion to leave the cell are activated and open. Potassium ions leave the cell. The loss of ions leads to a higher water potential inside the cell. Water leaves the cell by osmosis down the water potential gradient. The cells become less turgid or becomes flaccid. Gut cells shrink, causing the stomata to close. D1 talks about the effect of different concentrations of abscisic acid on root elongation. You are asked to describe the effect of abscisic acid treatment on the root elongation rate. In a description question, you should comment on the trend and provide data to support your statement. There is no mark for any explanation, so do not spend time doing that. In this case, there is a control and 3 other sets with different concentrations. You should make comparisons to discuss the effect. When you compare 0.1 micromole and the control, there is an increase in the rate. However, higher concentrations show the opposite effects. For 10 micromole, the rate stays roughly constant. It does not increase over time like the other 3 experiments. There is a mark for a data quote to support any of the points. You should compare the rates at 2 concentrations on one day for the first and second points. For the third point, you can quote the rates on 2 different days for 10 micromole. When you are quoting data, remember to place the points after the statement. This will show the examiner which marking point the data is for. 2 shows a passage about auxin. When auxin binds to the receptor, protons are pumped into the cell walls, reducing their pH. Expansins will then be activated. This enzyme disrupts the cross-link between the cellulose microfibrils in the cell wall. The loosened cell walls allow expansion to take place as water enters by osmosis. Question 4 is about GM potatoes. Table 4.1 shows the percentage of Colorado beetle larvae that die on the GM potato plant varieties and non-GM potato plants. A1 suggests what is meant by recombinant DNA technology. It is a technology that joins or combines DNA from different sources. You can also say that the DNA comes from different species. A few enzymes are involved in the procedure, such as restriction endonucleases and DNA ligase. Vectors are used to deliver genes of interest into the host cell. When a new gene is introduced to an organism, a GMO or transgenic organism is created. They created 2 different types of GM to compare the effectiveness of the 2 inserted genes. The results show a difference between BT and OC2 genes. The comparison allows us to see which genes works better at killing the larvae. The non-GM plant acts as a control. It is a baseline to be compared with the GM plants to show the effect of those genes. B. Discuss how the results provide information that could help to solve the global demand for food. Both A and B can kill the larvae. They are proven effective as almost all the larvae are killed after 72 hours. When pests are destroyed, crops would be healthier and less damaged. This leads to a greater productivity. Comparatively, A works better in destroying the larvae. It will provide more food, so it should be grown. 5A shows a few respiratory substrates and the oxygen needed for their respiration. You are asked to identify the substrate with the highest and lowest RQ values. RQ is the carbon dioxide produced divided by the volume of oxygen used when a substance is respired. In general, the molecules of CO2 produced are the carbon number of the respiratory substrate. When you divide the molecules of CO2 produced by the molecules of O2 used, malic acid has the highest value, while oleic acid has the lowest. B. Describe the similarities and differences between fermentation in mammals and yeast. Fermentation in mammals refers to lactate fermentation, while in yeast it is alcohol fermentation. In both cases, the pyruvate that is produced from glycolysis is used. The reaction occurs in the cytoplasm as mitochondria are not involved in anaerobic respiration. NAD is regenerated in the process. This allows the continuation of glycolysis as it requires NAD as a coenzyme for the dehydrogenase. In both situations, a redox reaction occurs. Pyruvate and ethanol are reduced, while reduced NAD is oxidized. The differences include the products of the reactions. They are lactate and ethanol. In mammals, it is a one-step reaction, as pyruvate is reduced to lactate directly. In yeast, it takes two steps. Pyruvate is decarboxylated to ethanol, and it is reduced to ethanol. Carbon dioxide is made in yeast via decarboxylation of pyruvate. It does not occur in mammals. It is reversible in mammals and irreversible in yeast. In mammals, lactate can be oxidized back to pyruvate. In yeast, ethanol is a waste product. Question 6 shows the genetic basis of the body color and stripes of the tiger barb. Table 6.1 shows the result of a genetic cross. A fish with a transparent phenotype was crossed with a tiger barb showing the albino phenotype to produce the F1 generation. These F1 offspring were crossed with each other, and the result is what we get in the table. A1 wants you to explain why the fish breeder concluded that the new transparent phenotype had occurred because of a mutation. An important concept is that all new alleles arise due to mutation. Since the transparent phenotype is not found in F1 but reappears in F2, it shows that the new allele is recessive. The dominant wild-type alleles marks it, so it was not shown in the F1 generation. It is inherited or passed down from one generation to another. It is not a phenotype that is caused by environmental factors. Here is a genetic diagram to help you to understand the cross better. You can refer back to this diagram for the following few questions. 2. The ratio is roughly 9331. To get the ratio, divide all the values by the smallest number and round them up. 3 wants you to explain the results in Table 6.1. You can refer to the genetic diagram I have drawn. Two genes control the phenotype, G for the color and B for the stripes. The genes are located on separate chromosomes. We know that they are not linked by looking at the F2 ratio. The autosomal link condition will not give us the 9331 ratio, as most offspring would have the same phenotype as the parents. Their phenotypes are not affected by their sex, so this is not a sex-linked condition. Each gene has two alleles. White stripes and transparent alleles are recessive. They cannot be expressed in the F1 generation in the presence of the gold and black stripes alleles. The wild-type alleles marks them. So, the wild-type is dominant for both genes. B. Describe the processes that occurred during meiosis in the F1 fish that allowed this variation to occur. Two events can lead to variation in meiosis: crossing over and independent assortment. However, since the genes are not linked, crossing over is irrelevant. It can only cause variation by breaking the linkage of genes that are found on the same chromosome. At prophase 1, homologous chromosomes pair up and form bivalents. They position themselves either way up on the equator of spindle fiber in metaphase 1. This leads to random segregation of the homologous chromosomes. This is the independent assortment. In our case, one chromosome of each pair has a dominant allele and one has a recessive allele. So, 4 combinations can be found in the gametes. They also give a mark if you use a diagram to show this. A1 outlines the similarities in structure between a neuromuscular junction and a cholinergic synapse. A neuromuscular junction is the point of contact between a motor neuron and a muscle fiber. Acetylcholine is released to stimulate muscle contraction. A cholinergic synapse is a junction between two neurons that use acetylcholine as the neurotransmitter. They have acetylcholine in vesicles. Exocytosis occurs to release the neurotransmitter into the synaptic cleft. Many events in a synapse require energy. For example, synthesis of neurotransmitters, active transport of substances, and exocytosis. So, they have many mitochondria to produce ATP. Both structures have presynaptic and postsynaptic membranes. These are the membranes before and after the synaptic cleft. The space between two neurons is called a synaptic cleft. The same term describes the junction between a neuron and a muscle. They have receptors for neurotransmitters on the postsynaptic membrane and the sarcolemma. The binding of acetylcholine to the receptors causes an action potential to be created. The presynaptic membrane for both has calcium ion channels. The influx of calcium ions stimulates the exocytosis of acetylcholine. On the other hand, the postsynaptic membrane has sodium ion channels, which allow depolarization to occur when sodium ions move into the cell. 2. Suggests why calcium phosphate formation in the sarcoplasmic reticulum may result in fewer power strokes occurring in sarcomeres. The formation of calcium phosphate decreases the amount of calcium ions and inorganic phosphate. So, we will answer this question by using their function in creating the power stroke. After the sarcoplasmic reticulum's depolarization, calcium ions diffuse into sarcoplasm. They bind to the troponin molecules, causing them to change shape. Tropomyosin will be shifted, and the myosin binding site will be exposed to actin. Exposure allows cross bridges to form. With fewer calcium ions, all these events will not occur as much. Inorganic phosphate is needed for ATP synthesis. Lacking it will cause a reduction in ATP production. Without ATP, cross bridges do not break. Myosin heads cannot reset to form the next cross bridge, which allows further contraction of the muscle fiber. In B, we have a diagram showing the cell signaling pathway of adrenaline. A is the enzyme activated by the G protein. It will then catalyze the formation of CAMP from ATP. It is adenylyl cyclase. The enzyme activated by CAMP is protein kinase A. It phosphorylates other proteins in the cell, leading to the desired cellular response. A wants you to identify a point where light intensity is acting as a limiting factor on the rate of photosynthesis. A limiting factor is a factor that directly affects the rates of reaction. It limits the maximum rate. The reaction rate is controlled by light intensity before the graph levels off. In this region, an increase in light intensity causes an increase in the rate of photosynthesis. So, putting an X at any point near the increasing part is correct. B discusses microarrays. They were used in an investigation to compare the gene expression of cow grown in high and low light conditions. The process is called transcription, when DNA is used as a template to synthesize mRNA. The opposite reaction is called reverse transcription. The enzyme involved is called reverse transcriptase. Transcription factors are proteins that control gene expressions in plants. They can help or prevent the action of RNA polymerase. 3. Suggest why more genes will switch on only in plants growing in high light intensity. In high light intensity, plants can photosynthesize more as more light energy is available for the light-dependent stage. Photosynthesis is an enzyme-controlled reaction, so the plant needs to produce more enzymes and other proteins involved in the process. For example, more rubisco must be produced for carbon fixation. This occurs in the Calvin cycle. More pigments are synthesized to protect cells from excess light. Some pigments absorb harmful light wavelengths. Enzymes needed to produce photosynthesis products, such as sucrose and amino acids, must also be synthesized in greater amounts. More proteins are synthesized to transport assimilates, such as sucrose. For example, the transport proteins involved in sucrose loading into the sieve tube element. There are 2 marks for AVP. You can get this mark if you provide suitable examples of the enzymes or proteins needed, such as electron carriers in ETC and ATP synthase for chemiosmosis. You can also provide details of how DELA may be broken down in high light conditions and how this leads to the expression of the genes. You can't get a mark for this question if you only state that more enzymes or proteins need to be produced without providing their specific names or functions. Question 9 shows an insect called green lacewings. A talks about its sense organs that detect sound. 1 wants you to outline how natural selection could have evolved the organ. You can consider how antibiotic resistance developed in bacteria through natural selection and use this concept to answer the question. First, a random mutation occurs. This introduces an allele that codes for a protein that allows some insects to detect high-frequency sounds. Best predation acts as a selection pressure. A selection pressure is an evolutionary force that causes a particular phenotype to be more favorable in certain environmental conditions. In this case, those that can detect high frequencies can survive as they can escape from bats more easily. They are said to have a selective advantage. These individuals survive, reproduce, and pass the beneficial alleles to the next generation. When this happens repeatedly, the mutated allele that benefits the insects increases in frequency. Eventually, more or even all the members of the population will have the allele, hence the organ that gives them the ability. 2. Describe the sequence of events that results in an action potential in a sensory neuron. Since the receptor is a cell and not part of a sensory neuron, there must be a synapse between the two. So, you should describe what happens in the synapse that leads to the action potential. The influx of calcium ions into the receptor cell leads to the creation of the generator potential. Calcium ions stimulate the vesicles containing the neurotransmitter to move towards and fuse with the presynaptic membrane. Acetylcholine or neurotransmitter is then released by exocytosis. It will then diffuse across the synaptic cleft down the concentration gradient. The neurotransmitter binds to the receptors on the sensory neuron due to their complementary shapes. The binding causes sodium ion channels to open. Sodium ions then enter the sensory neuron. This leads to the inside of the membrane becoming more positive. This causes the depolarization of the membrane of the sensory neuron. The action potential is created when the threshold potential is exceeded. An action potential is not created if the stimulation does not exceed the threshold. This is called the all or nothing response or principle. B shows a comparison of the characteristics of 2 species of green lacewing that evolved from a common ancestor. You are asked to suggest how speciation occurred to produce the 2 different species. There are 2 types of speciation, allopatric and sympatric speciation. Since they show overlapping distributions, it is more likely that they do not diverge due to geographical isolation. So, the sympatric speciation is a more possible mechanism. It may have started due to mutations. Different mutations occur in different members of the population, resulting in alterations to their alleles in various ways. The genetic differences lead to phenotypic or behavioral differences. This is evident by their different courtship songs and colors. The groups that show different phenotypes or behaviors are then isolated. This is called behavioral isolation or separation. Behavioral isolation refers to organisms that do not interbreed due to differences in their courtship or mating behaviors. Eventually, they lose the ability to reproduce together. They are now reproductively isolated. There will be no gene flow between them. You can also call this genetic isolation. Due to the lack of supporting evidence, allopatric speciation can be accepted as well. Allopatric speciation is always due to geographical separation. There may have been a physical barrier in the past that separated the population into different groups. Different groups have different mutations, resulting in changes to their alleles. They also experience different selection pressures as the environmental conditions are not the same in the two geographical regions. This change in the gene pool lead to phenotypic or behavioral differences. Even though the physical barrier is ultimately removed, the two groups are so distinct that they no longer interbreed. They are now genetically or reproductively isolated, having evolved into two different species. Question 10 requires you to outline the various ways in which genetic technology can be applied to medicine with specific reference to named diseases. Firstly, recombinant DNA technology can be used to produce proteins needed for treatment. It is done by introducing genes of interest into a host cell, such as bacteria. The genetically modified bacteria are then used to produce the proteins of interest. Then, provide examples of proteins that are produced and the diseases they are used to treat. Note that you cannot get the mark without naming the disease. Secondly, genetic screening can be done to identify the presence of disease-causing alleles in an individual. You can get marks by stating the alleles being tested and the disease they are associated with. If the diagnosis shows a positive result, early treatment can be done. Here, you can continue to discuss the application of genetic technology for genetic disorders, gene therapy. It is a treatment involving the insertion of a normal allele into the cells or tissues of the patient. If the allele is successfully expressed, the symptoms of the disease would be eliminated. Genetic disorders such as LCA and SCID can be treated this way. Note that this is not practical for conditions such as Huntington's disease that are characterized by dominant inheritance. There are two marks for AVP. For example, you can talk about xenotransplantation. It is a procedure that involves transplanting living cells, tissue, or organs from a non-human animal into a human recipient. GM animals are used to produce tissues or organs for transplantations. Additionally, scientists are now exploring the potential use of GM plants in vaccine production. They introduce genes that code for antigens into the plant's genome. The plant cells will then express the gene and contain the antigen. When they are eaten, an immune response will be stimulated in the consumer, achieving the goal of vaccination. If you know any other correct examples, you can also receive AVP marks. That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. Thank you for watching and see you again soon.